Hey everyone, welcome to Professor Long's Lectures in Anatomy and Physiology. I'm Professor Bob Long. If you're watching these videos, you know that we're in the coronavirus shutdown, and these videos are intended for use by students who are enrolled in my class at Del Mar College. The breadth, the depth, and the detail I go into is designed for my courses. If you're in another instructor's class at Del Mar or anywhere else in the world, the level of detail and the areas that y'all go into may vary. We all do things a little bit differently. So, if you find it helpful and you're not in my class, by all means use the videos. But again, the testing material that your instructor may cover may not include everything that I do and may include other things. Now, in the first video, this is the third video in our series on the blood. The first video was sort of an overview of the blood, plasma, formed elements. Second video we went into is we started to go into the red blood cells and what they are comprised of, hemoglobin and its structure, and erythropoiesis. Now we're going to go into white blood cells. So the technical name of white blood cells, the scientific name, is a leukocyte. And by the way, the C very often originally was spelled with a K. Leukocyte with a K is the old way to do it. I still utilize it that way sometimes, and some old textbooks have it that way. So if you see it in either form, it's the same thing. A leukocyte is a white blood cell. White blood cells stain differently than red blood cells, obviously, and they have a totally different function. The function of white blood cells is immunity. And immunity means defense against pathogens, disease-causing organisms, and foreign invaders, essentially. Things like bacteria, viruses, fungi, parasites, anything trying to get inside your body that doesn't belong is going to try to rob the nutrients from your cells, kill your cells, and steal their nutrients. And so our white blood cells are our immune system, our ability to fight off infections and diseases and things. Also, they can digest our own cells. If our own cells get worn out and need to be replaced, they get broken down, they can release chemical signals or get tagged, so white blood cells know to come get them. I'll show you what I'm talking about in a little bit. Now, there's a slight mistake in my note set. By the way, if you're following along in the note set, we're on page 39. I know I skipped page 38 in my videos. We're going to do it after this section because the definitions will make more sense once we've covered this. So, leukocytes are produced by a process called leukopoiesis. Let me just get this out of the way, okay? So, leukopoiesis is white blood cell production. We've covered erythropoiesis. Erythropoiesis is the production of red blood cells. Leukopoiesis is the production of white blood cells. The production of all blood cells is called hemopoiesis. It includes erythropoiesis and leukopoiesis. Now, when it comes to leukopoiesis, the production of white blood cells, a lot of this is directed by a class of hormones called thymosin. We've covered that on the endocrine system. Essentially, what we start off with is this, and I'm going to draw the chart going down, not across like I did for red blood cells. We start off with a cell called a hemocytoblast, which is the exact same cell that that red blood cells started off with. As a matter of fact, hemocytoblasts are a stem cell. They live in our marrow. These cells can start to turn off old genes and turn on new ones, start expressing different proteins. One of the things that hemocytoblasts can convert into is a pro-erythroblast. And I'm not going to go through these steps, but you know pro-erythroblast becomes an erythroblast, a reticulocyte, and then they become a red blood cell as they go through the process. We did that in the last video. The same hemocytoblast can become a leukoblast. The leukoblast is going to go through a series of differentiations and can result in 4 different cells as they develop. I'm not going to go into every step because it gets very complicated. But there's 4 major white blood cells that can come from this. There's one called neutrophils. And I'm not writing very neatly because I'm at the bottom of the board again. But they can develop into these cells called neutrophils, basophils, eosinophils, and the last one they can develop into is called a monocyte. So all of these cells come from the same stem cell that red blood cells come from. Again, there's a series of steps into becoming each one of these. But once it's developed into a leucoblast, it's almost completely dedicated to becoming one of these 4 cells. It's far more complex than this. The last thing that a hemocytoblast can evolve into is called a lymphoblast. A lymphoblast will become a lymphocyte. Now, these are the 5 white blood cells. This is leukopoiesis. A hemocytoblast differentiates into a leucoblast, which will eventually become either a neutrophil, or a basophil, or an eosinophil, or a monocyte. If the hemocytoblast matures into a cell called a lymphoblast, it turns on a totally different set of genes and eventually will develop into a lymphocyte. There are many more steps. There's some changes along the way that can occur. And if you take immunology, I'm sorry, it's a very tough class. But that's the basics of leukopoiesis. Okay? Now, which is just white blood cells. This would be our erythropoiesis, and altogether is hemopoiesis. Now I'm going to erase this, and we're going to talk about the 5 different white blood cells. And if you look in my note set, there is an error at the top of page 39. It says there are 5 major classes of white blood cells. And that's not true. There's only 2 major classes of white blood cells. There's 5 types of white blood cells that we listed, but they fall under 2 major categories. So the 2 major types of white blood cells are called granulocytes. Granulocytes are called granulocytes because their cytoplasm of these cells gets filled with these little grains or granules. They look like little dots filling the cell. They'll have a nucleus and a lot of cytoplasmic granules. Those granules are simply vesicles, bubbles of membrane, that have certain chemicals in them that make each one of the white blood cells slightly different. As we stain the cells, those granules and the cytoplasm all stain different colors. We wouldn't see these cells under a microscope. It would be like looking at saran wrap. You'd see right through them. So we take the cells and we put different dyes and stains, and the chemicals of those stains stick to certain chemicals inside the cells. Excuse me, I dropped my marker. So when we look at the granulocytes, they have lots of granules in their cytoplasm. I'm going to draw them out. This would be for lab also. The second major class is called agranulocytes. Agranulocytes, A meaning not or without. So, when it comes to the granulocytes, there's 3 types. One is called a basophil. Well, let's start with neutrophils. One is called a neutrophil. Now, the term phil at the end of the name means to love, like philosophy is the love of the sophistic method or thinking. Philanthropy, anthropos meaning man, so the love of man and the study of humankind, or the giving to humankind. When someone's a philanthropist, they give all their money away to help other people and things. Or a bibliophile, someone who likes books. So a neutrophil is a cell that likes this neutral staining dye. There's a dye that has a neutral pH. So they pick up this neutral pH stain. Now if I look at them, the cytoplasm of these cells usually stains a very light blue color. So when you look at them under a microscope, a neutrophil has a lot of light blue cytoplasm, but if you look at the nucleus, which usually stains a purple color, the nucleus isn't round like in many of our cells. The nucleus has many lobes. It'll have an enlarged area, it'll get real thin, it'll get thick again, and they can have all these thin areas and all these thick areas, and it stains a funky color. And no 2 neutrophils have the same shaped nucleus. As a matter of fact, if I draw a second neutrophil here, this one might look totally different. They can have lots of lobes, lots of enlarged areas. To me, they almost look like little sausages on a string. They get skinny and thick and skinny and thick and skinny and thick, but no 2 of them will look the same. The granules in their cytoplasm tend to pick up the same color of the stain that the cytoplasm is, but they're very thick and they're definitely noticeable if you have the right lighting on your microscope and the right strength of your microscope. So those would be neutrophils. Now when it comes to neutrophils, if I had red blood cells next to them, they're bigger than red blood cells. Okay, maybe not that big. If I were to put a red blood cell next to them, they're definitely bigger than red blood cells. Now the second major type of granulocyte we could look at is called a basophil, or let's look at eosinophils. Eosinophils tend to love this dye called eosin, and so they pick up an acidic stain, and it's red in color. So when we look at an eosinophil, eosinophils look about the same size as a neutrophil, but they tend to have what we call a bilobed nucleus. So we know neutrophils have multilobed nucleus. And some people call these cells a PMN. PMN stands for polymorphonucleocyte. And I know I'm getting close to the edge of the screen, so you may not be able to see exactly where I'm writing, but they can be called a polymorphonucleocyte. Poly meaning many, morpho meaning shape, and nucleus. Every one of them has a different shape, nucleus. Eosinophils, we know, have a bilobed nucleus. And I'm going to move the agranulocytes down because I need more room. So they tend to have a nucleus that has two lobes to it. I'm not going to fill it all the way in with purple. And the distinguishing characteristic under a microscope is that they have these very bright red granules that fill their cytoplasm. Okay? They just get filled with these red grains. And in these granules, we're going to talk about this in the next step, what's in the granules makes them behave differently. The third major class of granulocytes is called a basophil. Basophils, they like this basic dye or basic stain, and it tends to be a blue or a purple color, a very dark blue color. They are roughly the same size as neutrophils and eosinophils. They're bigger than a red blood cell. Their nucleus can take on different shapes, but you'll see them with these very dark staining cytoplasmic granules. I always think of it this way. If it's hard to see the granules, they're neutrophils. And they have a nucleus that's different. Eosinophils, the E, I think of the red in E for eosin, the red granules. And the B in basophil is for the blue granules, the dark purple or bluish granules. Now, if I look at the functions of these cells, neutrophils are the most abundant of all the white blood cells. There's more neutrophils than all the others, or at least it's thought so. Neutrophils, their granules, can release chemicals that attract other white blood cells to the injury site. So whenever you have an injury or you have an infection, when a neutrophil makes it to the location, it will not only start to attack the other cell, they are phagocytic. All three of these are capable of phagocytosis. But neutrophils will release chemicals that tell all the other white blood cells, hey, here's where the problem is. It's almost as if you sent in, let's say, a reconnaissance soldier, and they found where the bad guy's camp is, and they put mirrors so that all the jets and helicopters flying over can see where the bad guys are. Or if they dropped a smoke bomb and everyone knows where to attack. That's kind of what they do. They're usually the first one to the site. They release some chemicals that attract other cells to the location, and they are phagocytic. So if you're looking in the note set, it'll tell you everything I want you to know about these is in bold. They can phagocyte bacteria, and they release a lot of chemicals. Those chemicals happen to be called prostaglandins and leukotrienes. Those will attract other cells to the location and also dilate red blood vessels in the area. Now, eosinophils are much lower in number. Eosinophils, their cytoplasmic granules can release chemicals called well, not a lot of histamines, but they release some chemicals that can increase inflammation in an area. And some of those granules have cytotoxic enzymes that are toxic to other cells, And literally, as they spit these enzymes out, it will eat holes in the other cell's membrane. Almost like if someone grabbed a bad guy and started stabbing them, and it would release all their contents and kill them. So it tells you this in the note set. Did I leave eosinophils out? Oh no, they're down here. Eosinophils take on an acidic dye. They are phagocytic, and they can definitely attack parasites. They're one of the ones that really increases in number when we have a parasitic infection. Now, one of the things that happens is this. Just like in most countries, if you're not at war, you decrease the size of your military to conserve expenses and energy. When you go to war, you have to increase the size of the military. You know you're going to lose soldiers, sadly, so you have to recruit a whole bunch. The same happens with white blood cells. All of the white blood cell numbers are kind of low. That's why there's so few of them. If you have some kind of infectious agent, then the number of white blood cells can go up. When they get chemical signals like prostaglandins and leukotrienes from neutrophils, the other cells not only get attracted to the site, they start to undergo massive amounts of mitosis and increase their numbers so that they can attack the infectious agent. It turns out that eosinophils release a lot of acids and enzymes that eat other cells and particularly go up in number in parasitic infections. Okay. Finally, there's basophils. The granules in basophils have two compounds in them. Those compounds are called histamine and heparin. So I'm going to talk about basophils and histamine and heparin. Heparin is an anticoagulant. So an anticoagulant prevents clotting, at least temporarily. This can prevent clotting. So let's say you cut your skin and you're bleeding. One of the first things you want to do is you want to flush the wound out and wash any bacteria out or any infectious agent. Second thing you want to do is you don't want it to clot right away. It does take a few minutes. But you want all the white blood cells. With the dilated blood vessel, more white blood cells and your blood can be shifted to the site, and the white blood cells will increase in number and they will all make it to the site of infection. So there's a chemical reaction that occurs that dilates the blood vessels, which increases the redness and the swelling, more fluid, more swollen, and then attracts all the other white blood cells. And heparin prevents clotting of that so that some of the white blood cells can make it into the wound. Eventually the clotting reaction will occur, but it takes a little bit of time. The second compound that is released is called histamine. Histamine is a vasodilator. And what that means is it will dilate blood vessels in the area. And as the blood vessel dilates, that means increased blood flow, increased redness with more red blood cells and white blood cells, and increased swelling. They also make some of the blood vessels a little bit more permeable so white blood cells can exit the bloodstream and attack the infectious agent. So you need to know that these release prostaglandins and leukotrienes that attract all the other white blood cells to the site and can increase inflammation. And they are phagocytic. They can attack antibody-coated cells. Eosinophils are also phagocytic, but they can release cytotoxic enzymes and acids that kill other cells. And basophils are filled with histamine and heparin. Heparin is an anticoagulant so that we flood out or wash out the wound, and other white blood cells can make it there. And then histamine is a vasodilator. By the way, basophils and eosinophils go up in number when we have allergic reactions to things. When you breathe in cat dander from a cat, if you're allergic to cats, or some kind of tree pollen, when it enters your system, these cells detect these foreign substances and release their chemicals. And the histamines cause the blood vessels to dilate, increasing fluid, everything gets impacted, and your eyes water, your nose gets stuffy, and you get all the discomfort that you get with an allergic reaction, like seasonal allergies. And you take a drug called an antihistamine, which will allow the blood vessels to constrict and decrease all the swelling and inflammation. So an allergic reaction is immune response with swelling, vasodilation, and the movement of these white blood cells into the site. And in particular, in immune reactions, both basophils and eosinophils can go up in number. I think that's what I want you to know about that. There is a type of allergic reaction that we call eosinophilic asthma. There's a type of asthma where people's respiratory tract will constrict and they can't breathe to prevent you from breathing in debris and foreign substances. And eosinophils can be activated in a type of eosinophilic asthma, which cause a massive overreaction and swelling. And you can read up about that. I'm not going to test you on it. What I want you to know is that these are the three major classes of granulocytes, or the only three granulocytes. You need to know what colors do they detect or pick up when we stain them. You need to know these have a multi-lobe nucleus and pick up cytoplasmic granules that stain the same color, a sort of a light bluish color. Eosinophils pick up red granules and have a bilobe nucleus, and basophils pick up a bluish purple granule, stain I should say, and these are the chemicals that they release, histamine and heparin. Know that, okay? That I got a little off track, not exactly the way that I wanted to work, but nonetheless. Now we're done with the granulocytes. Let's look at the agranulocytes, okay? So I'm going to erase all this so we can focus on the agranulocytes. Essentially what you need to know if you're in my class for testing purposes is the wording that's in bold under each one of these cells in the note set, okay? That's what I was trying to get across to you. I feel like I was stumbling a little bit. The second major class of leukocytes is called agranulocytes. There are two subtypes of agranulocytes. One is called a monocyte. Monocytes, if I were to put them next to the other white blood cells. So let's draw a red blood cell just for reference. Let's draw a neutrophil just for reference. Neutrophils, eosinophils, and basophils are all roughly the same size. This one has a multi-lobe nucleus and it's going to pick up these light granules. Now if I were to draw a monocyte is much bigger than both of these. They're the largest of all the white blood cells. They usually have a C or U-shaped nucleus and a very thick one at that. They actually have some little granules in their cytoplasm, but they're so tiny that they didn't show up on microscopes, so they got labeled as agranulocytes. Monocytes are highly phagocytic. They will attack anything that gets in their way. If it's a foreign cell, they gobble it up, okay? Now, one of the things I did not tell you yet is this. If I were to draw a blood vessel here, almost all of the white blood cells that we've talked about, actually all four of them, neutrophils, eosinophils, basophils, and monocytes, live in the bloodstream. They spend almost all their time in the bloodstream amongst all the red blood cells. The only time they exit the bloodstream is if I, let's say, have a cut and I have some foreign cells get in, like a bacteria or a virus or something, then the white blood cells can actually migrate out of the bloodstream. They'll squeeze in some openings here and pop out into the periphery and start eliminating and attacking the foreign agents. All four of the cells we've talked about so far are phagocytic, and monocytes are highly phagocytic. They just gobble stuff up. They're filled with granules that have enzymes. They're lysosomes, essentially. And eosinophils and basophils, they'll all move out and do their thing. Now, there are some monocytes that actually migrate out and live in the peripheral tissues. They live there for the rest of their lives. It turns out that we call those macrophages. And some books will refer to them as a tissue macrophage. It was once thought that these were two totally different cells. Turns out that monocytes are macrophages that live in peripheral tissues. If they're in the bloodstream, we call it a monocyte. If they live in a peripheral tissue, we call them a macrophage. You've heard of a few of these macrophages before. In the nervous system, in our brain and spinal cord, we call them microglia. In bone cells, they're called osteoclasts. And in liver cells, in the liver, we call them Kupffer cells. K-U-P-F-F-E-R. Kupffer cells, I think there's one F. In the liver, it might be two F's, I think it's two. I haven't looked at that word in a long time. But Kupffer cells live in the liver. I actually have a whole list of these. If you look on page 40 of the note set, I've listed a bunch of them, but these are the three that I really want you to know. There's also mesangial cells and other cells in other tissues. But these are all macrophages, osteoclasts, microglia, and Kupffer cells of the liver. They're all highly phagocytic, and in the liver, they're going to be recycling red blood cells like crazy. A lot of monocytes also live in lymph nodes, but we're going to talk about those in a little bit. Now, I'm done with macrophages for now. Know that they're highly phagocytic. Inside the bloodstream, they're called monocytes. In the peripheral tissues, they're called macrophages. Know the three subtypes. Finally, the last of the agranulocytes are called lymphocytes. Once I'm done talking about lymphocytes, I'm going to go back. I'm going to stop the video because we're probably getting close to 30 minutes if we haven't surpassed it. And we're going to do the definitions, and we'll do blood clotting. So, monocytes and lymphocytes are agranulocytes. Now, one of the things that we know about lymphocytes is they're the smallest in size of all the white blood cells. So if I had a red blood cell here, let's say that this represents a neutrophil and this represents a monocyte. Okay? The neutrophil actually would be a little bit bigger than the red blood cell. Lymphocytes are about the size of the red blood cell, only smaller. And they're almost entirely nucleus. It's like a giant nucleus with just a little bit of cytoplasm. So lymphocytes are the smallest of the white blood cells, and they are almost entirely nucleus. There's different types of lymphocytes that I want to cover. Let me check the time on my video. Good, we still got some time. So let me talk about the different classes of lymphocytes. Now, one of the things that's in our note set is this. There's two different types of immunity that we're going to talk about. There's called specific immunity or acquired immunity, and then there's non-specific immunity, also called innate immunity. All of the white blood cells that we've talked about, except for the lymphocytes, are part of our innate immunity. Innate immunity. The nate comes from nature, which means birth. If you speak French, naissance means birth, like the Renaissance is the rebirth. This means inborn. Innate, also called, I'm drawing a blank, I just said it. Well, innate immunity is also called non-specific immunity. Sorry, I'm not going to quit on a 26-minute video. This type of immunity is done by all the white blood cells except for lymphocytes. When you are born, you have monocytes, neutrophils, basophils, and eosinophils that will attack anything that gets in. It doesn't have to be specific. If a bacterium gets in, they'll all attack it. If a virus gets in, they all attack it. Okay? Lymphocytes, on the other hand, participate in what we call specific immunity. Specific immunity is also called acquired immunity. This is immunity that you gain only after you're exposed to the foreign substance. You're born with lymphocytes, but they're not activated, they're not dedicated to fighting infections. They're just cruising around. Now, once you get exposed to a specific antigen, a foreign invader, some lymphocytes can be dedicated to fighting only that specific invader. The other lymphocytes are not involved. If a different type of cell tries to invade you, let's say one's a bacterium and the other one's a virus, when another class of lymphocytes, which have not been dedicated to fighting something, for example, if I have two lymphocytes here, if I have a bacterium that gets into the body and this lymphocyte starts to attack that bacterium, it becomes dedicated to fighting only that bacterium. If I have a virus that invades the body and this lymphocyte is going to attack that virus, for the rest of its life, it'll only recognize that virus. It will not attack that bacterium, and this lymphocyte will not attack that virus. They get dedicated to fighting a specific foreign invader, only after you've been exposed to that invader. So you have to get exposed to things to develop immunity against it, okay? So now, knowing that, we're going to talk about the different types of lymphocytes and how they function, okay? So I'm going to erase all of this. I feel like this isn't the smoothest lecture I've ever given on all of this, but nonetheless, it is what it is. I hope you're learning from it. I'm going to erase all of this too, and we're going to talk about lymphocytes and specific immunity. So now, if I take a lymphocyte, if I can look inside the nucleus of this lymphocyte, there are strands of DNA called chromosomes. And let's say there's a particular section of this that's going to give us an antibody. Antibodies are the proteins that can bind to foreign substances. This particular gene, this region on the chromosome that develops an antibody, is going to make 4 different protein chains. 2 of the protein chains are very large, and they call them heavy chains. 2 of the protein chains are very small, and they call them light chains. They get stuck together with some bonds called disulfide bonds, but they look like these little Y-shaped molecules. Now, if I were to analyze the amino acid sequence of this antibody, it turns out that most of the amino acids in this are called fixed region. Okay? The fixed region means that the order of the amino acids never varies from one antibody to the next. Okay? They also call the fixed region the non-variable region because it does not vary. But if I look at these little tips here, that area is called the variable region of the antibody, which means the amino acid sequence from one antibody to the next can vary. It can be a different amino acid sequence. And without getting into real specifics, and I did a lot of antibody work in my graduate training. We developed monoclonal antibodies to specific proteins we were trying to isolate. So I have a really strong understanding of the immune system, but it's so complex we can't go into great detail. Suffice it to say this. Essentially what happens is when a lymphocyte, which has not been dedicated to fighting an invader, encounters some foreign cell, and that cell has a protein on its surface that does not look the same as the proteins on our cells. Our cells might have all of these surface proteins, but this one particular protein here is unique. And our lymphocytes and our cells in our body don't have the gene to make that protein. When this lymphocyte encounters that foreign protein is called an antigen because it will generate an antibody response. It will cause lymphocytes to make antibodies against it. When this lymphocyte encounters this foreign cell, it literally kind of stops and says, hey, what is this? It takes a while. And it has the ability with some enzymes to cut up this gene and rearrange the gene sequence, which will arrange the amino acid sequence in the variable region. And eventually, it will spit out an antibody that's going to be able to bind to and stick to that particular protein. Now, I tend to draw my antibodies as little Ys, but really they're shaped more like this. There's a unique three-dimensional shape. And if I have an antibody that makes this particular, if I have a lymphocyte that makes that specific antibody, then it's going to spit out tons of these antibodies. And not only that, the lymphocyte will undergo rapid mitosis and clone its genetics so that every version of itself is going to be able to also secrete the exact same antibodies. So it's going to flood our bloodstream with thousands of these and billions of these molecules or these antibodies. Now the reason that's important is this. If I have a foreign invader, usually more than one gets into the body. And all of these foreign invaders are going to have these same proteins on their cell membrane. And they usually have a lot more than one copy of this protein. So I'm going to draw a bunch of these foreign cells here with this foreign protein on the surface. Now what happens is, if all of these antibodies can recognize this, one antibody can bind to this cell and another cell that has that protein in it. Another antibody will bind this one and that one. Another antibody will bind these two. Another antibody will bind these two. Another antibody can bind these two. And essentially the antibodies can start to clump all these foreign invaders together in one particular location. And we call that an antibody labeled antigen, because the antibody has stuck to it. Once these lymphocytes make that specific antibody against that specific foreign invader, they can't make any more antibodies. Now that all the immune cells, all the foreign invaders are in one location, what if I brought in a monocyte and he starts engulfing these? Several monocytes and eosinophils and basophils and neutrophils can come in and attack and remove all the infection at once. This usually takes a few days to a few weeks. So if you do get sick, sometimes like when you get a viral infection, you have to go through the 2-week infection before your body clears it. If your body can't clear it, if it's a particularly bad virus, then hopefully we have medication, antivirals, or other ways to try to fight it, like the coronavirus and other viruses, okay? So that's essentially what's happening in specific immunity. Now, I'm getting close to the end of my time. My videos only run about 33 minutes, and I'm past it, so I'm going to keep going. Essentially what happens, for those of you that aren't aware, after 33 minutes, my camera shuts off and starts immediately a new video. Since I'm already past it, I've already started the second video, I'm going to finish up lymphocytes, okay? So this is specific immunity. Once a lymphocyte encounters a foreign invader, it makes an antibody against some surface protein called an antigen, and then that lymphocyte is dedicated to that. Now, watch. Once we've eradicated the infectious agent, the number of white blood cells that I need that are going to secrete that antibody can go back down to a bare minimum. And the count of antibodies in our body will go down, but we still have them. And if we encounter that foreign agent a second time, and it hasn't mutated, then this lymphocyte will remember that. We call them memory cells, B memory cells. The memory cells remember the infection. The second time it enters your body, you wipe it out before it even causes any problems. Like when you get the chicken pox, you get it once. When you get certain infections, you get it once and then you develop immunity to it. The only time that doesn't really work is if the virus, there's a type of virus called retrovirus. They actually have an RNA genome. They don't use DNA, they use RNA as their genetic material. When they infect the body, when they infect a cell, they inject their RNA. The RNA has to be converted into DNA. It's called reverse transcriptase is the enzyme that does it. And because of that copying process doesn't have any correction mechanism, they make mistakes, and those mistakes result in genetic variation, or what we call mutations. So for example, the flu virus does this. When you get the flu, you develop immunity to it. The next year, if the flu virus has mutated, you don't have immunity to the new strain, so now you have to get a different vaccination. And that's why sometimes the vaccines aren't always 100% effective. They're pretty effective. I get my flu vaccine every year, and I'm not trying to tell you what you should do, but for me, I do it so that I don't get the flu, and it's been pretty successful in my lifetime. Now, that is exactly how we take advantage of vaccines. I don't know if you know this, but I'm going to give you some information. Not all of this is testable material, but I think it's important for you to know for your future. The term vaca means cow, right? So a vaccine comes from the fact that when there was a disease called smallpox infecting the world and wiping out most of the population of Europe, scientists noticed that the milk maidens that would go and gather milk from the cows would pick up the cowpox. Cowpox is a virus that's of the same class of smallpox. And the pox are pustules on the surface, and they would see them on the cows, and the milk maidens would get the cowpox virus. They would develop immunity to a certain part of the protein in cowpox. And then if they got exposed to smallpox, the smallpox virus had a similar protein, and so they had immunity against that, and they never got sick. And so this scientist actually created the first vaccine from vaca for cows. He would take some of the pus from a cow and infect himself, and he didn't get smallpox. And as he did it to his family and other people, he actually developed the vaccine to smallpox using cowpox. Now we use the term vaccine. And what they didn't know is what's happening here. Once the smallpox virus, you developed immunity to it, The next time it invaded your body, you wiped it out before you got sick again. It didn't mutate a lot. Now, let's say a different foreign agent enters your body, and it has a totally different protein on its surface. It's got some of the same ones that our cells have, but now it's got a unique protein. This lymphocyte is already dedicated to fighting something else. So a different lymphocyte, a new one, that's never been exposed to any of these foreign antigens, will recognize this foreign antigen. It'll do the same thing. It'll rearrange its DNA, so to speak, and then it'll make a body against this protein. So this white blood cell, this lymphocyte, will specifically fight off this foreign invader. This lymphocyte will fight off this foreign invader, so we call it specific immunity. Now, one of the things we know is, what if I just could isolate the particular protein, or I could take, say, for example, the virus that makes this protein, but I could kill its DNA. I can treat it with a chemical that disrupts the DNA and makes it a non-functioning live virus. It's a live virus, but it can't replicate itself. If I could inject that into someone's body, and their immune system can make antibodies against that protein, then when the real virus invades your body, and it has that same protein, your body will already be immune to it. We call that immunization or vaccines. So we purposely expose our immune system to the foreign antigen, this protein in the surface, and then we develop immunity against it, so when the real thing invades your body, you wipe it out. For most viruses, that works really, really well. It doesn't work well for viruses that mutate rapidly. For example, HIV, human immunodeficiency virus that leads to AIDS, or that's why we haven't been able to create a vaccine to it, and the flu. We can create vaccines to different strains of the flu because we know in advance when it's coming across the ocean. But for the most part, you have to get a new one each year. But flu is still somewhat stable. The coronavirus is also a single-stranded RNA virus, which means that it can mutate, and it's been around for generations in animals. It finally mutated to where it infects human lungs, and that's all we need to say about that. So now we know about specific immunity, we know a little bit about lymphocytes. Now here's what I might test you on, okay? I need you to know that lymphocytes are not part of the acquired or non-specific immunity. I mean, they're not part of innate or non-specific. They are specific or acquired immunity. They're a type of acquired immunity. You only acquire it after exposure. There's different types of lymphocytes, and I want to list them here. They're in a table in my note set. And I'm not going to cover all of them. I'm going to cover all of them but one. B cells are cells that can secrete antibodies. They release those proteins against a specific antigen. Let me switch markers. Okay? And once you're infected, some of those B cells can stick around for a long time and become what we call B memory cells. There's another type of lymphocyte called T cells. These are all different types of lymphocytes. This is one of the things some of the nursing faculty and some of the students that do the immunology in the nursing program told me they need to cover. The T cells come under different classes. So for T cells, there's one called a helper T cell. Helper T cells help the immune process, and one of the things that they do is that they release chemicals called cytokines. Cytokines are some chemicals that attract other white blood cells to the site and attract other lymphocytes to the site. So they help the process by saying, hey, here's where the bad guys are. Okay, that's pretty much what I want you to know about them. There's another one called a natural killer T cell. Natural killer T cells kill infected cells, and usually virally infected cells and tumor cells. When our cells start to develop into a tumor, like a cancerous tumor, they express different proteins. And when these cells recognize it, they will attack those tumor cells. So they're tumor suppressor cells or tumor fighting cells, and they can fight cells that are infected with viruses. And when helper T cells release cytokines, those cytokines can also bind to some of these foreign cells and let these guys know who to kill. Okay. And finally, there's cytotoxic T cells. Cytotoxic means they are toxic to other cells. And cytotoxic T cells, they can release acids and enzymes that lyse infected cells. These cells are not phagocytic, but they can bind to other cells and poke holes in them and essentially help kill these virally infected and tumor cells. So if you look in my note set, I think on the bottom of page 40, it talks about the different types of cells. The natural killer cells, you don't need to know the percentages of them. The T helper cells and cytotoxic T cells and B cells. You don't need to worry about the gamma and delta T cells. We're not going to talk about them, okay? So this is what I want you to know about the white blood cells and the immune system. I hope that all makes sense to you. I hope you learned something. I felt like this wasn't my smoothest lecture. It was a little herky-jerky. But nonetheless, I hope you had as much fun as I did. I hope you learned something. I hope you have a little bit of understanding of our immune system and how vaccines work, because they're going to be important for you to understand when you become a healthcare professional. This is something that my students come back all the time and say, we really need help with this, Mr. Long, and I got to explain it. We could go into greater detail, but immunology is in and of itself an entire 16-week course and could be taught over 2 semesters. It's a lot of information. If you ever have to take it, I'm sorry. Anyway, hope you had as much fun as I did. I hope you learned something. I hope to see you on the next video. Thanks for watching.